Welcome to the last session of the uh, Immunology special of Samyojana. And today we'll have the last two presentations. First, as you can see on the slide, is by Rachna Gaulikar on the immune system as a therapeutic agent, and a very nice concluding article by Srishti. So without any further delay, Rachna, please go ahead and present your story.
Thank you, sir. Hello, everyone. I'm Rachna, and I'll be presenting an article from Scientific American, The Immune System as a Therapeutic Agent, which is a special issue from Life, Death, and Immune System. So, and the author of this paper is Hans Wigzell. So, let us just have a brief out on the author. So, Dr. Wigzell is the president of Karolinska Institute, general director of National Bacteriological Lab in Stockholm, and also a member of Royal Academy of Sciences. So, he was featured in one of the movie called Odyssey of Life. So, Odyssey of Life is in which it opens up with the ultimate journey, which shows how millions of years of evolutionary history are reflected in life before birth in species such as humans, chickens, and fish. So now, the main focus of the paper is new technologies and insights into the molecular underpinnings of the immune system provide the basis for novel approaches to vaccines and other therapies. So, in this paper, we'll be discussing more about the vaccines and the therapies that led to the further research in the scientific community. Now, the immune system is an alchemist's dream. So, uh, to see an alchemist in your dream is a sign that you desire to get new information. And if you use it cleverly, you will be able to demonstrate your potential fully. So, its multifaceted methods of attacking such intruders, such as bacteria or viruses, they offer an abundance of compounds and molecular strategies which extend the scope of medical intervention. So, I am talking about the immune system. And then, using our knowledge of the immune system, scientists or researchers can actually strengthen its response uh, to give to a given antigen, direct it to combat and overlook, and in cases such as rejections or misguided attacks. So when there are successful attempts which give rise to a kind of golden therapies to prevent and to treat disease. So moving on to the next, let us begin with the innovative means of limiting immune system responses. So our efforts are currently underway to improve exciting vaccines to make them more specific and to eliminate any adverse reactions that may accompany immunization. So the refinements and a thorough understanding of certain illness have suggested innovative means of limiting immune system responses. In cases like allergy, autoimmunity, organ rejection by pinpointing and blocking the action of specific molecules. And also in contrast to other therapies are being crafted to increase attacks on cancer and AIDS. Perhaps the most exciting is creative use of components of the immune system as biological mimics. So remember the biological mimics because we'll be discussing about this in the further slides as enzymes that can help in the treatment of disease. So now, in general, there are two approaches to harnessing the immune system therapeutically. So not surprisingly, these approaches reflect two aspects of immunologic function. The first one is antigen specific and the second is not antigen specific. So in the antigen specific, the T cells, they respond to a precise target, that is a foreign organism or an entity. So the T cell form and action is called cell mediated and the antibody response is termed as humoral. You might be knowing about this from your before presentations that were presented in the last few sessions. And what is not antigen specific? This type of response brings about the actions of compounds known as cytokines, which resemble hormones or antibacterial peptides. So, therapeutic uses of antigen specific strategy are familiar because they include one of the most medicine's potent defenses, vaccines against the infectious diseases. The key to vaccines lies in the immune system's remarkable memory. And not only can it recognize and destroy nearly any intruder, but the immune system also remember for decades. For example, when a person is given a weakened dose of an infectious agent, such as the virus that causes polio, for example, we are considering a polio, uh, which is injected into a person's body, and when their body responds by making antibodies, and the T cells will ultimately eliminate the virus. So once the organism has been obliterated, the responsible antibodies and the T cells remain alert, and they protect the immunized individual against any later or perhaps more virulent encounters with the same organism. So now, as so, vaccines of various kinds have been used for thousands and hundreds of have been used for hundreds of years. So, there's a short timeline of events in the history of vaccine development, and let us go through the timeline of vaccines. So, in AD 23 to 79, Roman scholar Pliny the Elder suggests that livers of mad dogs could offer protection against rabies. I was kind of very interested on this part. Like at that time, they were thinking in a way that how mad dogs could offer protection against rabies. And then in 1500s, Asian physicians used smallpox crust as form of immunization with mixed success. That means here they were actually giving the smallpox crust to inhale and then look for the immunization. And in 1796, the British physician Jenner uses cowpox virus to vaccinate effectively against smallpox. You might all be knowing about the Edward Jenner's discovery. And in 1885, Pasteur develops a rabies vaccine, finally. And look at the timeline of the years from 1823 to 79 to 1885. It took these many years to develop a rabies vaccine. And from 1890 to 1904, Von Behring and Kitasato Shibasaburo successfully test vaccines for diphtheria and tetanus. And this tetanus vaccine was made available in 1914. And the typhoid and cholera vaccines were produced in 1900s, and tuberculosis vaccine was made in 1920s. So moving on to the further timeline of vaccines, you could see there are many, right? So in 1930s, diphtheria and yellow fever vaccines were produced. In 1940s, influenza and whooping cough vaccines were created, and then and there was again one of the discovery from Salk and Sabin, who discovered different forms of poliomyelitis vaccine. So in further slides, you will get to see historical picture where people stand in a queue to get this vaccine. In 1960s, measles and rubella vaccines were made, and in 19 and also mumps vaccines were designed in 1960s. In 1970s, meningococcal diseases and chickenpox vaccines are produced, and also the pulmonary diseases vaccines is also developed during 1970s. And finally, in 1980s, hepatitis B and a combination vaccine for measles, mumps, and rubella became available. And for this vaccine to become available, there were a few wonderful experiments that were discussed in the paper. And in 1990, the vaccine for Haemophilus influenzae, a cause for meningitis, is introduced. So the timeline that was shared in the study was only till 1990, because this paper was published somewhere around 1993, and which was very exciting to read. So here, as I told, that this is one of the images where the Harlem residents who formed a queue to get vaccinated at a mobile clinic in 1959. And this unit was a part of health department drive to vaccinate everyone in New York City. And at that time, by 1959, the smallpox was eradicated, and WHO was focusing on poliovirus. So now let us move on to the effective ways to administer immunization. So, developments in the past decades have revived such investigations. And using molecular techniques, scientists can now rapidly locate the genetic component of a microorganism that give rise to an illness, as everyone know that. So, they can isolate the protein or a series of proteins which arises from those genes and manufacture it in a pure form and in a great quantity, and then vaccinate people with specific element rather than the entire organism. And furthermore, if any proteins in a potential vaccine are found to be harmful, they can now be easily deleted or modified using the molecular techniques. So the effective ways were that the specificity increases the effectiveness of a vaccine. Monoclonal antibodies can be used to discover the neutralizing determinant or a vulnerable portion of a bacterium or virus. So, and then a large majority of antibodies that the body produces to conquer an organism. For example, an influenza virus is ineffectual because the target proteins that do not allow them to eradicate the organism. But the remaining minority of antibodies are focused on a weakness in the viral defenses. And now, ways to obtain a killer T cell response. So, for instance, um, a killer or a cytotoxic T cell cannot attack an antigen unless it has been taken inside a cell and presented on a surface compound called the MHC, that is major histocompatibility complex. And in the past, ensuring about this absorption and subsequent that the vaccine can be made of live organism or virus or sometimes a dangerous proposition. So here the new adjuvants are the substances that are used in the preparation of vaccines and which can circumvent this problem because they can ensure that the antigen will be seen by the correct immune system cell. And probably the researchers can insert an antigen into the liposomes or tiny capsules that are made of fatty molecules. So once the liposomes are in the body, the macrophages devour them. So like waiters bringing dinner to ravenous diners. So the macrophages and the other immune system cells that have consumed the antigen-laden liposomes and themselves surrounded by the T cells. And there is another fascinating way of obtaining a killer T cell response is that while injecting a DNA that encodes a foreign protein into the skeletal muscle. So here they have considered skeletal muscle because it seems that the muscle cells, which are much larger than most of the body cells, can serve as factories where antigens are produced, processed, and presented on the MHC molecules. And then the muscle cells make the foreign protein from the injected DNA and then offer it up as antigen that is recognizable to the T cells. So although there is an autoimmune response against the muscle cells, which is negligible. So despite these improvements in vaccines, many microorganisms maintain their capacity to outwit the immune system. So including the malaria parasite continuously alters its appearance and rescrambles its genetic system. So although we have a more complete understanding of the structure and of these organisms or the parasites, which will try to improve vaccine design, but according to the study that they were discussing, the pathogens will always exist that they are capable of resisting the immunizations. So now, how are vaccines developed to treat IgE-dependent allergies? Might be in the last presentations, you might know what is an IgE-dependent allergies. So here, the vaccines that were discussed till the last slides were that the Dr. Han have described in the paper are prophylactic vaccines. So the prophylactic means that they are given before contact with a disease-causing organism. So now let us move with how our vaccines developed to treat Ig-dependent allergies. Vaccines that may serve vaccines that may serve as remedies develop develop and have been developed and have, like their cousins, benefited from the recent advances in molecular biology. So the first one is immunotherapy. So immunotherapy for patients who have allergies to um, to pollen or animal proteins, for instance, is transforming. So, the traditional treatment of this problem involves the repeated administration of an allergen. So, for example, there was an experiment that was conducted using a cat hair in highly dilute or gradually increasing concentrations. They used the cat hair uh, with the person who is allergic to cat hairs, and such experiments, like such classical therapies, reduces a person's sensitivity to the allergen, but it works in only some patients. Otherwise it may completely make the experiment upside down if the person is not able to uh, like suffer with the cat hairs. So, a more sophisticated alternative to vaccine therapy would attempt to disarm the antibodies involved in the reaction. And the form of immediate allergy described in the above experiment is usually caused by the overproduction of antibodies or immunoglobulins belonging to a class called IgE. So, where there is overproduction of antibodies, and I could quite relate with this experiment. So, thus, a logical treatment would suppress IgE and Ig related reactions. So, it is known that two chemical messengers, that is interleukin 4 and interleukin 5, produced by T cells augment the production of IgE. So, the potential success of this approach has been suggested in laboratory experiments on mice. Rodents which were unable to produce interleukin 4 are also unable to make IgE. This is kind of interesting, right? So, and there is another chemical messenger, that is gamma interferon, brings about the production of molecules that inhibits the formation of interleukin 4 and interleukin 5. So finally, after the two chemical messengers that gave their properties, therefore the vaccines that developed to treat Ig dependent allergies could include an agent that would lead to the production of gamma interferon. So there is an image through which we can understand about how the chemical messengers produced by the immune system, such as the gamma interferons. The gamma interferons are depicted as purple spheres over here, these, and then which can be used to increase the attacks on the antigen. So before an antigen can be seen by T cells, it must be processed by a macrophage and presented on the MHC, that is major histocompatibility complex molecule. And the gamma interferon amplifies this process by causing macrophages to display more antigen, thereby enhancing the T cell activity. And here comes the secret to treat autoimmune disorders and prevent organ transplant rejection. And the key for this is curbing the immune system. So the goal in transplantation is to create specific tolerance in the recipient towards the antigens from the donor, or it could be in case of a bone marrow graft, to deter the lymphocytes produced in the donor marrow from attacking the recipient. There is a well-known example. We all know that, but just to mention, we know that this option is within reach in humans because non-identical twins who shared blood circulation during pregnancy can accept tissue grafted from each other. So that's what the main goal of the transplantation tells us about. And then a spectacular means to achieve tolerance would be to harness an autoimmune response. So the recipient immune system could identify and then wipe out the T cells carrying receptors for transplanted antigens. And then it has indeed been possible to induce the autoimmunity against the T cells with a specific antigen binding capacity. So there were few experiments that had been led by Dr. Wigzell, the author of this paper. So Dr. Wigzell has been involved with several experiments in which rats from strain A were injected with T cells, that sort of antigens from the tissues of rats from strain B. I hope you're getting my point, that how Dr. Wigzell have been through these experiments. And when they injected the two strains, when they injected from the two strains, they found that in a minority of animals, the vaccination brought about lifelong specific tolerance against the B antigens, and there were no any complications further. So these exciting experiments have, however, been very frustrating because it proved that impossible to make these autoimmune systems produce specific tolerance in reproducible manner. So, and yet the fact that the experiments resulted in low but specific number of animals becoming functionally tolerant, which illustrates the biological validity of the concept. Even though in the reproducible manner, the experiments weren't having a specific tolerance, but biologically they could illustrate that there is something in this concept which could lead us to the further research. And then the most successful immune intervention for transplantation directs the monoclonal antibodies against the cell mediated part of the immune system. So, here the first the monoclonal antibodies to be registered as a drug is targeted against the constant part of the antigen binding receptor on the T lymphocytes. Now, uh, the the constant part of the antigen binding receptor, which are on the T lymphocytes, now that causes the rejection of a kidney transplant. So, unfortunately, although the drug reduces the rejection of kidney, it has a significant disadvantage because it blocks the T cell functions. And apart from blocking the T cell functions, uh, these play more, I mean, these also play a major role in other things as well, and you will get to know in further slides. So monoclonal antibodies with more selective immunosuppressive activity are being designed. So some are directed against CD4 or CD8 molecules of the cell that bind with antigens, and others against adhesion molecules on the T cell surface, and still against the receptors of interleukins. Um, According to the reports that were shared in the study, several of these antibodies are beneficial when they were used in the preliminary trials in the patients, and still they are not sure about this study because it is still in the trial and error method. So here you can actually see an image which has an advanced the especially the laboratory technology, which has advanced dramatically in the past decade, so thereby permitting the development of more selective and sophisticated vaccines. So in this photograph, you can see a researcher who is using an automated computer control equipment to produce monoclonal antibodies. And I wondered, at that time they had such good technologies that they were using for such experiments. And moving on to next, with vaccines have the potential to thwart attacks on the cells. So in addition to block the immune system attacks on foreign antigens, vaccines also have this potential role that they play to attack on cells. So here, the causes of many autoimmune diseases, which includes rheumatoid arthritis, insulin-dependent diabetes, and multiple sclerosis. So in rheumatoid arthritis, the T cells attack the joints. In insulin-dependent diabetes, the T cells attack insulin-making pancreatic cells. And in multiple sclerosis, the T cells attack the fatty sheets around nerve cells. Um, and probably this is not known, but they have these assumptions that they make. So, and after that, the animal model suggests that immunization with self components can induce autoimmune disease. For example, there was this experiment done by injection of a rat collagen, which can give rise to rheumatoid arthritis like disorder in the same animal. But uh, but then it was unclear whether these models reflected what occurs in humans. So now that there is enough information out there to know about autoimmune disorders to permit vaccine research. We can be more specific to go with the antigen specific therapy for autoimmune disease that has only two possible targets. So the tactic here is essentially to use autoimmunity to combat autoimmunity. And it's sounding so simple, but it is very difficult to do that. And when immunologists examine the local T cell populations causing a disease, they often find an enormous enrichment of T cells that recognize a certain antigen at the site of the illness. So, as there are only two possible targets, the disease inducing agent or the antigen, and the other is the antigen specific lymphocytes or T cells that are attacking the tissue. And most of the autoimmune diseases are cell mediated. For example, here Dr. Hans, the author of the paper, and his colleagues have seen that for an autoimmune lung disorder called sarcoidosis, is a disease that is characterized by growth of tiny collection of inflammatory cells, that is granulomas in any part of your body, most commonly the lungs. Most commonly the lungs and lymph nodes, but it can also affect your eyes, skin, heart, and other organs as well. But this research tells us that 100% of the patients of similar genetic background have usually large number of T cells with a characteristic receptor in their lungs. So now, in cases of autoimmunity, graft rejection, and allergy, the immune system reaction needs to be scaled down and redirected. So the opposite is true for AIDS and for cancer also. So here, the immune system attacks need to be scaled up. And the desperate situation brought about by the HIV, which causes AIDS, has prompted attempts to develop therapeutic vaccines, in addition to ongoing efforts to design a preventive vaccines. And HIV did play a role for the further development in the therapeutic vaccine designs. So there are two attempts that they made. So the first attempt involved isolating the envelope proteins called GP120 or GP160. Here they used the researchers, they isolated GP160. GP160 is a combination of GP120 and GP41 from the HIV. So the protein is given to an HIV infected individuals in which in whom the symptoms are not developed yet. And as initially shown, as the researchers, they isolated this GP160 and injected into the asymptomatic HIV patient, the procedure the procedure that is generated, which enhance the immune response against the HIV. So this reaction includes the increased titers of antibodies that neutralize the envelope protein and other HIV variants. So here, and this experiment was done at Walter Reed Army Institute of Research, and it was further confirmed at Karolinska Institute, where the author actually worked. And normally the levels of CD4 T cells, which bind to the HIV, drop in infected patients, but although this observation need to be confirmed while they were doing this research, so there has been at least two reports that tells that CD4 T cell decline is curbed following vaccination with GP160 protein. So that is why here the researchers isolated GP160 protein and then injected into the asymptomatic HIV patient. And so there is another second interesting candidate for therapeutic HIV vaccine was based on autoimmunity. So in the series of the experiments, this was performed at Harvard University by Norman Letwin and his group. And they injected monkeys with recombinant CD4 molecules in order to block the infection. And further, they found that the animals made antibodies against CD4. So these antibodies, they appear to have no effect on normal CD4 T cell function, but yet they inhibited the HIV and SIV. So you might be wondering, what is SIV? So SI, so the so here the green monkey virus, usually described as simian immunodeficiency virus, and from the replication in vitro, and probably the antibodies that work because they disturb the viral binding site on the cells and thereby prevent the virus from binding and entering the CD4 T cells. So if the virus cannot gain entry, it cannot destroy the cells. So further immunization with the CD4, which are the recombinant molecules, in the in the in the green monkey virus, that is SIV infected monkeys, caused a reduction or a disappearance of virus from the blood for up to 4 months, and this lasted for like 4 months. So after that, so that wasn't sufficient to stop the research there, and probably they had another promising line of attack. It has to exist so that the further research could lead. So immunologists have been able to elicit both the humoral and cell mediated responses using hybrid molecules, that is the artificial structures that combine the features of other immunologically relevant molecules. And probably you will get to see in the next slide how this actually worked. So one interesting hybrid consists of an anti-tumor antibody and a super antigen. So super antigens are proteins of mostly microbial in origin that have a unique ability to activate a substantial proportion of T lymphocytes regardless of their antigen specificity. So, in the treatment of cancers, which employs two aspects of immune response, the antibody recognition of an antigen and the T cell. So, the T cell reaction to the super antigens. So, super antigens are microbial, as I already mentioned, which are attacked by many T cells. So, therapeutically, the strategies which fuses an antibody recognizing a tumor antigen with a super antigen to form a hybrid, as you can see, the hybrid molecule, uh, and and then the antibody, and when the hybrid molecule meets the tumor, the antibody portion binds to the antigen and super antigen attracts the T cells. So the T cells carry out their function to destroy the super antigen and kill the tumor in the process. So this strategy is effective because T cells are more destructive than antibodies. So now, antibodies can be used to mimic biological substances and to curb immune responses. You might be wondering that how you might be wondering that how antibodies can curb immune responses, right? So here, now let us jump into a few of the history of how antibodies mimic the biological substances. So, in addition to protecting against the diseases, antibodies can be converted into highly potent antigens, which are capable of provoking a strong immune response. So, in 1960s, Jacques Uddin, he is from Pasteur Institute, as you can see in the image, which showed he showed that some antibodies could be recognized by other antibodies, and those other antibodies are further recognized as antigens, and he called those antigens as idiotypic, and the antibodies that reacted with them which reacted with them, he called them as anti-idiotypic. So the concept of antibodies and anti-antibodies was further extended or further researched by uh, Nobel laureate Niels K. John into a vision of immunologic networks in such a system. So some anti-idiotypic antibodies react with the antigen binding site of the idiotypic antibody and look the antigen itself. So, and then they can therefore foster active and protective immunity against infectious diseases. And also it has now been shown that the anti-idiotypic antibodies may indeed function like the antigen that they mimic. So let us have a clear understanding by the research that they conducted. So antibodies may even be designed to take over functions of antigens or antigenic molecules. So one of the first reports that were expanded the function of the antibody responses was from the experiments which were done on the insulin antibodies. So these are the insulin antibodies you can see in the image. So the immune system creates the antibodies to insulin. So the red color ones are the antibodies which the immune system created. And such antibodies can be harvested in the laboratories, and they are used to provoke the production of antibodies. So the purple ones are used to are used for the provoking of the production of antibodies. And when they introduced into a patient, like the antibodies are isolated and then injected into a patient, the antibodies, they block the antibodies that harm insulin. So they also bind the insulin receptor and cause blood glucose levels to drop. So in other words, some of the antibodies, they function like insulin. So with this, so these were like the last experiments that the study have conducted about the therapeutic opportunities and how vaccines work and what are the mechanisms of the antibodies and all. So with this, I had few conclusions. So although the predominant approach to designing immunotherapies has been based on antigen specific elements, as we have been mostly talking about the antigen specific throughout the presentation of the immune system, where the antibodies and the T cells, that picture is rapidly changing. And the antigen non-specific parts, as I mentioned in the beginning of the presentation, the antigen non-specific parts of the immune system have resulted in a novel channel for medical investigations. So again, after a series of over-enthusiastic claims by researchers, interferons are now finding their place as more conventional drugs to treat some viral diseases, among the chronic hepatitis B and C, larynx papilloma, and relatively rare tumors such as hairy cell leukemias and carcinoids of the gut. And the other conclusions are the discovery of new forms of treatment, such as those that include cytokines, shows us that the human immune system has riches as yet unearthed. So, the complexity of this defensive system promises a wealth of proteins and molecular strategies that can continue to forge medical innovation. And I have like had a very brief conclusions over here, but I would tell you all to just go back to this paper and have a read, and then you will probably understand how cytokines and how the interferons led their experiments to these conclusions. And like miners equipped with brighter headlights and sharper picks, immunologists can select and extract these treasures with increasing ease. So with this, I would like to conclude my presentation, and thank you for listening. I'm done with the presentation. Am I audible?
Yes, you are. You are. I hope my screen is visible.
Yes. Yes, Srishti, your screen is visible.
Thank you. Hello, everyone. I'm Srishti Chiple, and today I'll be presenting a very interesting Scientific American article titled Will We Survive by Avrion Mitchison. Avrion Mitchison is a scientific director of DRFZ in Berlin Mitchison read zoology as an undergraduate at Magdalen College, Oxford, where his tutor was Sir Peter B. Madaber. He did research, lectured, and taught at Edinburgh University, Harvard University, Stanford University, and University College London, where from 1971 until 1991 he was professor of zoology and headed that department. Mitchison has received many honors, including the Paul Ehrlich Prize and the Scientific Medal of Zoological Society of London. He has published over 200 research papers. All right. So over the week, we have seen how immune system fights off the microbes and protects our body. In our conversation till now, we assumed our body and its immune system on one side of the picture and microbes on the other side. But in reality, all of these species, including us, survive under the same umbrella of life. We are adapted to each other in one or the other way. However, from over a century, which is very trivial amount of time from evolutionary perspective, we have been trying to live in an artificial environment free of microbes. Although this might sound good and obvious, there is a possibility that this underemployment affects our defense mechanism. Our current lifestyle has increased our chances of getting exposed to a variety of microbes. A virus now exists that attacks the very defense on which we rely for survival, the human immunodeficiency virus or the HIV. The Legionella bacterium, the Lyme disease spirochete, and Rift Valley fever virus also exemplify nature's ability to challenge the immune system radically. Even old pathogens invent new tricks. Thus, the question arises, will Homo sapiens and microbes continue to coexist, or will one side win? This is a very clear question asked by the article. Will we survive? Will Homo sapiens and microbes continue to coexist, or will one side win? I feel that the best answer to this question is we don't know, because of our inability to look into a larger view and to look through the future. So, one thing that can possibly be done is looking through the past. The author has done the same in this paper. He enlists some facts and experiments from the evolutionary history of our immune system and thus tries to understand its feature. The immune system has always served the sole purpose of defense against infection. No external factor has shaped it. Let's take the example of bubble babies. This is a genetic condition called adenosine deaminase deficient severe combined immunodeficiency or ADA deficient SCID, often called the bubble baby disease. Children born with SCID must be kept in controlled, isolated environments. The condition can, if untreated, be fatal within the first year of life. So they are kept in a very controlled, sterile environment called a bubble. Now, some scientists propose that immune system stimulates the growth of red blood cells. So this should mean that the bubble babies of their and their animal counterparts, mice with a congenital thymus defect, for example, should have an erythrocyte count lesser than normal. However, this is not the case. They produce normal number of antibodies. Some other workers have proposed that the immune system can prevent some forms of sterility by warding off the effects of paternal leukocytes. Yet, mice that have a congenital thymus defect, if kept alive in a semi-sterile environment, they produce quite well. Now, this does not mean that we can't make the immune system perform these and other functions. However, it is not easy to persuade the system to perform chores to which it is not accustomed during the course of evolution. Another major feature of the immune system is that it has evolved through a process of elaboration. It overrides the ancient mechanisms and replaces it with something better. Like the defenses that protect invertebrates are wandering phagocytic cells, as well as proteins floating freely in blood fluids that can bind to invading bacteria. These defenses can perform most tasks of the full-fledged immune system, except for mounting heightened responses against organisms that are previous invaders. The ancient defenses have no specific memory, which now is a hallmark of the immune system proper. The immune system also shares some similarities with the nervous system in its evolutionary process, but unfortunately, we cannot trace most of the evolutionary steps that the immune system took. Virtually all the crucial developments seem to have happened at an early stage of the vertebrate evolution, which is poorly represented in fossil record and from which a few species survive. For immunoglobulins, however, we can discern much of an evolutionary story. Presumably, the first system comprised a single immunoglobulin producing gene. It quickly recreated itself, establishing a series of duplicates, each making a different immunoglobulin molecule. Then control mechanisms emerged that could direct the production of separated gene segments that were able to recombine. Yet, even humans retain in a corner of their immune system something of the primitive arrangement. The genes that control the production of the lambda light chain immunoglobulin, one form of which contains only two members. Recent research on the immune system of fish and amphibians has identified another interesting survival. These cold blooded vertebrates have relatively slow acting immune systems equipped with poorly diversified antigen receptors. The tadpole of the common frog, for example, makes only about 100 different antibody molecules. On the other hand, frogs have potent defense peptides called macronins that punch holes in the bacterial cell wall. The immune system then probably passed much of its early life in a sluggish, relatively inefficient style, playing a fairly minor role in the defense. The system came fully into its own only much later on, after revolution of warm-bloodedness enabled our cells to multiply rapidly. As the immune system grew in importance and complexity, it underwent a series of internal reorganizations, which involved compromises or trade-off of one kind or the other. Now, the body does not have unlimited metabolic resources. The investment of these resources thus needs to be balanced in a regulatory system necessary for keeping the entire enterprise under control. Thus, in human immune system, there are more regulatory T cells known as the CD4 cells than any other kind. The B cells also furiously compete with one another for tiny amount of antigen needed for survival. The winners are assured the reward of massive replication. Cells that do not find antigen die. Where nature fails to provide evidence to support the inquiry into evolution, immunologists have borrowed a tool from economists, that is, computer modeling. Franco Senda of Hospital for Joint Diseases in New York City and Philip E. Seaton of IBM Thomas J. Watson Research Center have formulated a cellular automaton for closely modeling the cellular events that take place within the immune system. This technique can answer many significant and deep questions. We can now begin to derive some answers to our question. I would like to do so by dispelling a possible misconception. So far, this account of evolution of immune system might suggest that process had mostly played out itself long ago. In fact, the reverse is true. The human immune system is almost certainly now evolving more rapidly from generation to generation than ever before. Our immune system is intensely polymorphic, that is, occurring in several different forms, in particular with reference to species or genetic variation. Polymorphism is indeed complex and more interesting than simple selection in favor of immune responses to parasites. The reverse also occurs. A vigorous process in all likelihood creates a rich variety of genes whose product suppresses the immune response. Such genes are committed to limiting an immune reaction that directly damages the body in course of responding to a pathogen. They suppress friendly fire, so to speak. Nowhere is the balance between the two kinds of control displayed better than in leprosy. The disease afflicts more than 1 million people in the tropics throughout the world. It disappeared from Europe only in the past few centuries for reasons that are unknown. Leprosy, a disease no longer endemic to Europe, shows a range of the pathology that is the result of immune system's ability to shut itself down when the danger exists that a response will damage the host. In the patient ad left, Genes are expressed that have led to a lack of effective response. Consequently, leprosy bacillus multiplies and collects in fluid-filled blebs on the skin. Shared and unshared. Now, Richard Dawkins from University of Oxford has noted that relationship between pathogen and host can range from complete compatibility to fatal divergence, depending on the extent to which they share their agendas for survival. When a pathogen's genome travels in host gametes, the two share a common agenda and thus agree that the host should be healthy and capable of reproduction. The gametes of many mammal species, including raccoon, carry a significant complement of such benign and even useful viral genes. The relationship between rabies virus and its host illustrates the opposite effect. The virus, which spreads through contact with saliva, has little interest in host survival. The historical trend in the relationship between humans and their pathogens, the author believes, has been toward the shared agenda. Can we benefit that? Are we confident that this tradition of live and let live can persist in modern world? Well, opportunities have never been greater for mixing hosts and parasites that have no previous expression of one another. About 1 billion human beings join the world's population each decade. Many of them crowd into the mega cities of developing countries. High speed aircraft transportation facilities, the easy movement of millions of people around the globe. Recent past overs illustrations of what can happen when people meet a pathogen from which neither their evolution nor their immune system protects them. One need only recall the English treaty about the malaria infested west coast of Africa. The Gulf of Benin, where few comes out, though many go in. Has the immune system then reached its apogee after a few million hundred years it has taken to develop? Can it respond in the time to new evolutionary challenges? These perfectly proper questions lack sure answers because we are in an utterly unprecedented situation. Even if we knew how often in the past host species has been wiped out by their parasites, that knowledge would tell us little about ourselves as we stand at present. Yet, there are grounds for optimism. Never before have we succeeded in eradicating a disease simply by enhancing the activity of the immune system, as was done with smallpox less than 20 years ago, which would, of course, be a good example. Never before has science has been so sophisticated and so solidly lined up against infections, thanks to the WHO and more particularly its special research programs, and to such national organizations as Centers for Disease Control in Atlanta. But above all, we should put our trust in powers of microevolution and in the capacity of polymorphism to pull new defenses out of its vast storeroom and inability of host and parasite to ultimately to adopt a common agenda. But that's all on my side. Thank you so much for listening.
Thank you very much, Srishti. And I had some brief issue with my network a little when Rachna was finishing up. But on the whole, it was very interesting because uh, especially today's two articles, listening to it, it didn't feel like we're listening to something from 30 years ago, almost 30 years ago. And I think this series itself, I think, is very interesting. I did not think of it's almost like it's been engineered, right? So right now with the whole pandemic going on and so much discussion about the immune system, I did not. Uh, somebody might think it's actually very planned. The very plan is for the reasons of the pandemic, but it turns out actually it was completely independent of that. But it seems very apt. So on the whole, I think very nice presentations, Rachna and Srishti. We will have a little more discussion later on. But if there are any questions from the audience? Okay, I will take that as a no from the audience, and we will. So this is. I think this series has been fantastic. I really enjoyed all of the sessions, and today's especially was a nice conclusion to the discussions going on in the past. It was like I felt, at least they were definitely very specific uh, details, scientific details, but it felt more of a philosophical conclusion than very objective scientific conclusions. Uh, and I think I am also convinced that these short, focused, special editions or special sessions might be a good way to kind of delve into the past and or maybe into very focused areas of studies. So with that, I will formally close, and we'll have, as usual, an informal conversation. Uh, I mean, very soon in the next few minutes. So I'm again assuming that there are no further comments or suggestions from the audience, and we will uh, hopefully meet again in a similar session. I mean, a series of talks and series of articles soon enough, and I'm looking forward to more participation. So that will be kind of a formal closure to this series on or the special on immunology.
Sir,
Thanks everyone for
I would.
uh,
I don't want to come suggest, but I want to comment. Should I?
please go ahead. Yes, please go ahead.
So, first of all, I think that vaccine is the most said word in this 2021, and even getting to know more about it was very interesting. So, coming first with the Rachna's presentation, it was very nice and precise. And also at the end, when she concluded her presentation, it was the best part because she presented about a paper. So her presentation was a conclusion or the summary or the recap for the paper. And even she gave a precise conclusion for her own PPT. So that indicates that she really worked on it. So it was great presentation from Rachna. And now coming to Srushti. Srushti's presentation and everything she told was more like a YouTube video where the YouTuber himself considers that the audience, they don't know anything about it, and she tried to give a from the bottom line to everything about the topic. So it was great session from both of you. Thank you so
Thank
much.
you, and it means a lot from somebody who hasn't been following the rest of the talks in the series, and also who is not a regular in biology discussions, at least from our perspective. So that, I mean, the fact that these two talks engaged you as well speaks volumes. Thanks again,